Okay, good morning. Okay, good morning, everyone. Thank you for coming today to today's Ido Golding's talk. I hope the people online can also hear as well. If you do, just say yes on the chat, so we have no technical issues. So today I will let Sylvain Gadon introduce Ido, and then we can start.
Hello, everybody. So it's a pleasure to have Ido Golding visiting us. So he's coming from the US. So he's a professor in Illinois University. So Ido was trained as a physicist. So he did his PhD in Israel. And then he spent a few years in Princeton learning molecular biology and learning also about phage biology. And today he's going to tell us about the tools that he's using to look at the behavior of viruses. So he's looking at single-cell biology to explore the life history of viruses, and in particular in phage lambda. Ido?
Merci. Hello, everybody. So some of you attended Marianne's seminar yesterday, and I want to continue her advocacy of bacteriophages, viruses that infect bacteria, as an excellent entry point or starting point for quantitative and predictive biology. Just one sort of maybe technical or pedagogical comment, because again, maybe I'm coming from a background that is, or disciplinary background that is different from some of yours. So if I'm not being clear, just stop me and ask, or if you think I'm saying something wrong, you should also stop me. And again, coming from physics, I grew up in the tradition of a seminar being an ongoing dialogue, not a monologue. So if I don't make it through my slide, that is okay. Now the question that we are focused on is the question of dormancy, of viral dormancy in phages, and how we can use it to test our predictive power. Okay, so the viral dormancy is probably something that, something you probably know is very common in all kinds of viruses. Of course, HIV does that, and herpes virus does that, and countless other, not all, but many viruses do that. And likewise for bacteriophages, viruses that infect bacteria. And the model system that we use to look into that question is arguably the best characterized biological system, bar none, and that is bacteriophage lambda. So that is a virus that infects E. coli bacteria. Let me decide which of the tools I want to use. Let me try this one. Okay. So the virus arrives and lands on the surface of the E. coli bacterium, injects its DNA, and then a decision is made, a binary decision between two possible outcomes. By default, what the virus does is what viruses do best. He replicates as rapidly as he can, produces, let's say under lab conditions, 100 or 1,000 viral particles. Within an hour, they kill the cell and they go out to infect more cells. However, there's an alternative path in which the virus goes dormant. So it shuts off all the virulent functions and integrates its genome into the host genome. And so then its replication is only passive as part of the bacterial growth and division. Okay. Now, some of you may know that that is not me. A lot of the language in phage biology comes from Greek through French, and so the terms are like that. And so lysis is the viral virulent path, and lysogeny is the dormant path. Now, the dormant state is very stable. It can be maintained for millions of cell generations. At the same time, it's not infinitely stable. It is only what we would call it metastable, because in response to the right signal, the virus senses danger to the host cell and switches very efficiently back to the lytic cycle, and then again replicates and kills the cell. So a very simple question to pose is, can we predict when dormancy will be chosen? In a given infection event, can we predict what will be the outcome? And again, as a physicist, my single test for do I understand anything is predictive power, right? I understand gravity, right? Because if you tell me what's the height of the apple on the tree, I can tell you how long it takes to hit the ground and what speed it will hit the ground, right? So I understand gravity, okay, as a physicist. Likewise, here's a very simple case because it's a binary choice. Can I predict what it will be? Okay, so that is one way from the point of view of physics to ask, do I understand dormancy lysology? Now, let's start with sort of maybe an optimistic indicator for ability to predict. And this is experimental data from my lab, and it's really practically a modern redoing of a classical experiment by Philippe Kurilsky in 1973. And what we have is the following. So the x-axis here is the multiplicity of infection. Okay, how many viruses co-infect, copies of virus co-infect a single cell. And in this case, this is just obtained by mixing different ratios of bacteria and phages. The y-axis is how many lysogens, dormant cells, cells with dormant phages did I get? And this is just obtained by plating on antibiotic selection because we've engineered our phages so they have an antibiotic resistance marker in. And the data, different colors, different students over 20 years in the lab. There's even probably data from my days as a postdoc here. And you can see that they all lie on a very reasonable trend. And you can also see that here's a model that appears to capture the data quite well. Okay. Now, what is that model, that mathematical model? The model is very simple. The model says, in the tube, I have random collisions of phages, viruses, and bacteria. If a single virus infects a cell, the result will be cell death, okay, not dormancy. But if two or more viruses simultaneously infect the cell, I will get lysology. And this is that prediction, okay? One way of directly intuiting this, if you look at this slope in logarithmic scale, you get a slope of 2. And that tells you you need 2 phages simultaneously to get lysology, okay? So there's a simple model. It seems to capture the data quite well. And the other nice thing about this model is if you come from the, let's say, ecological point of view, you can make a very clear rationale out of it because you would say, well, what the phages are doing are they're inferring the relative abundance of phages and cell. Because if phages come in the cell and there's 2 or more of them co-infecting, that suggests that there's many more viruses than cells in the population. And so making 1,000 new viruses, killing the cell and going out looking for targets is futile, does not make sense, okay, if you want to say it that way. And so therefore, dormancy should be chosen, okay? So that is the rationale. And as some of you know, there are other viruses and phages that use different alternative strategies to try to sense that same information. What is the relative abundance of viruses and hosts in the environment? Okay, so this is a good start and very promising. Okay, but despite now it's over 7 years of studying lambda, this really best characterized system, there's really multiple elements of this decision, this counting by the phages that we don't understand, and I'll show you some examples of that. Now, one of the aspects that makes it complicated and makes it hard to understand is the element of viral replication. Okay, so one way to think about it, and we often look into that, is the in-host dynamics of the virus. Okay, so when we said if a single virus infects, the outcome is viral replication, lysis. Okay, so that means you go from 1 copy to 2 to 4 to 8, and then a few hundreds, and you go and you kill the cell. Now, as we said, if you start with 2 phages, you have to end up with a single phage going in the genome. Right? So there's sort of an inversion of the copy number. You go from 1 to many or from multiple to 1. Okay? So how that happens, because that means that along the way, for example, there's always going to be, let's say, 2 or 4 viruses. How does the viral circuit identify that the 2 or 4 is the initial number, in which case it has to respond in dormancy, or this is just ongoing replication that should proceed and result in cell death? And of course, this in biology, it's not that there's a decision and then there's replication. This is all going on at the same time. The virus comes in, immediately begins replicating. During that replication, the genetic decision is made, oh, should I stop and go dormant, or should I go on and make as many as I can? So this is quite confusing how that all works at the same time. So that is one of the things we started looking at to try to make sense of. Now, let me tell you just a bit about the types of experiments. And experiments are always the the meeting place of what you want to do and what you can do, right? And so one of the things we can do is we can simultaneously detect inside the bacterial cell the genome of the phages and one or several of their mRNAs, the expressed mRNA. So not going into detail, but genetically we engineer the phages so they have a binding site for a fluorescent protein, so that in the cell, this is one E. coli cell, so it's about a micron. Here's the scale. I have 1, 2, 3, 4 phage genomes. So 4 phages got into the cell, and I'm imaging them. And then I'm labeling 2 of the key regulators of the decision, the C1 gene that promotes dormancy. Here are the copies of mRNA, and the CRO gene, this is the mRNA that promotes lysis. Okay. So already from this first picture, you can see something that maybe is hints of something interesting. The 2 or 3 of the viruses are expressing the dormancy genes. One is expressing the lytic promoting gene, the viral gene. Okay. So we perform an infection. These are chemically fixed cells. So at different times, we can measure the number of copies of viruses and the expression of some of these key genes. Now, there's sort of compromises or choices we have to make. So at this level now, we can do better. But at this level, when we did this, we couldn't count as many different mRNAs as we want. It was easy to do 3. So we're looking at 3 genes that are, this is definitely not all the genetic network, but it's believed to be the core of the genetic network. There's these 2 antagonistic transcription factors, and there's an activator of the dormancy gene. Okay? So we have a core network, definitely not all of it. The other element is that we thought, well, replication is so complicated. Let's start with non-replicating viruses. Let's only look at the decision circuit and not worry for now about replication. So that means that we began by using a non-replicating mutant of the phage. So you'll see me calling it P minus. P is one of the genes involved in replication. So we mutate that gene. And so the phage has all the genetic circuitry for the decision making, just not replication. That also makes it simple that when I count a number of copies of the phage in the cell, 3, 4, 10, it's the number that came in. I don't have to worry about its history for now. So then we can write a simple, ODE model that captures these regulations, but of course we don't know the parameters. We would need to parameterize them, and that kind of model captures the experimental data very reasonably, not perfectly, but very reasonably. So here we have experimental data. So these are times after infection. And this is the copy number of the mRNA average in each cell. So we have C1, that is the driver of dormancy. We have CRO, that is the driver of lysis, and C2 is an activator of this guy. And the different colors are shades are different multiplicity of infection. Okay, so this is a cell with all the cells with 1 phage, 2, 3, 4, 5. So the model pretty well captures the data. Now it's not magic because we had 3 parameters, and so it's not, but it's still encouraging. And the dynamics, both the measured and the inferred, are very consistent with what we know about the genetic networks in terms of the order of the promoters turning on and everything. I don't have much into that. But one thing that jumped at us when we did that, we said, okay, the model's working, the data is nice, everything is good. The problem is there is really no sign of a diverging cell phase. Right? What we expected to see is MY1, single phage, you go one way, MY2 or more, you go the other way. But you can see this is just a scaling of the same curve. Okay, in fact, if I divide the curve for C1 by the multiplicity, they collapse on each other. Okay? So the system responds to the multiplicity, but it doesn't make a bifurcation or a divergence of a decision. So that was very odd. And then we realized, so one thing we could do is, because now we have a model, this is RNA, you can say, well, let me look at the protein level, which we were unable to measure experimentally, but now we have the model that we inferred, we can use that, and we can look at the trajectories in the protein space of the 2 regulators. The C1 is the transcription factor driving dormancy, and CRO is a good proxy for the virulent behavior. And you can see the trajectories are all the same. Okay, I mean, they scale in magnitude, but they all have the same shape. There's no topological difference between them. And so you can decide on where you would put a threshold for choosing one phage or another, but you cannot choose any threshold for this behavior that says, oh, I go from lysis to lysogeny, from virulent to dormancy when I increase the multiplicity. the topology doesn't support it. All you can infer is that when I cross some specific multiplicity, I will get dormancy, which is what happens in experiment, but I just never reach the lysis behavior, the threshold for replicating and killing the cell. So what is the problem? So we said, well, what happens if we do include replication? And turns out that including replication in the model solves this problem. Okay, so we take our model, this is the genetic circuit, but now we include the regulation of the viral copy number. Okay, so again, we don't know a lot, we don't know all about it, but we know some of the regulation, what activates or represses the replication. And we can again calibrate the model by experimental data. So here we're measuring the average viral copy number in the mutant, which of course nothing happens. And in the case of lytic reproduction of the wild type, so it gets to a few hundred copies in an hour. When you recalibrate, you take the same genetic circuit, but now with the presence of the replication you put in the model, then suddenly you see what we sort of expected to see. You have a very clear divergence or bifurcation. MY1, single phase infection, explodes into the lytic viral behavior. Higher multiplicities, you turn right and you go to dormancy. Okay. So the model says, yeah, that's all you need, and that will give you the behavior. We still, now we have the model, we use the model to look at things that you cannot look experimentally. And we use the model to ask, how does the replication actually enable the decision instead of like obscuring it? Okay. So very briefly, the main thing we found from the model is that the response to the multiplicity, the counting, if you will, that happens in a well-defined time window. Okay, so the viruses come in, and for the first, this depends on very much on the experimental condition. For the first about 20 minutes, they all do the same thing. They just express this gene C2, and that leads to the activation of the gene C1. And so at the end of these 20 minutes, what you get is an amount of these proteins C1 in the cell that's proportional to the multiplicity. So this C1 gene or the protein product is what counts. If I, after 20 minutes, ask, or 30 minutes in this simulation, how many copies of phages came in? The answer is measure how much C1 protein you have, calibrate, and normalize, and you know how many phages got in. Once that window of counting takes place, then there's no more counting. The system stops responding to the multiplicity, to the copy number. That's why it doesn't get blinded, confused by replication. And so I don't want to go too much into this, but you can ask, when I have two phages, if they come simultaneously, I know what the outcome is. But when phages come with some intervals, what is the impact of the temporal interval between incoming phages? And the answer is, if they both come in within what we consider the time window of responsiveness, they count as two. If they come in at the end of that window or later, they count as one. So in other words, there's a well-defined time window during which counting takes place of multiplicity. After that, doesn't matter. You can come in, you're not being counted anymore to the phage. And so the replication on one hand is what is required to even get lytic behavior. Okay, so in a non-replicating phage, you just never reach sufficient protein product to make to choose the lytic outcome. On the other hand, when you have replication, in terms of lysogeny, two phages are enough. Okay, so if you have a non-replicating phage, then at the end of the counting period, if you have two phages, you still did not have enough gene product to choose dormancy. So one of the other consequences, there's two consequences here. One, non-replicating virus cannot even choose the viral phage. The other thing is, it requires more phages to choose dormancy. And that is something that can be nicely validated in those Kurilsky-style measurements by looking at the slope of lysogeny versus multiplicity. So when you use a non-replicating phage, two phages are not enough to give you dormancy. You need 3 or 4. Okay. So with that as sort of a broader angle to look at it, it tells us that when I look at the fate diagram of the virus, I already have to consider one more axis. So one was multiplicity of infection, but I already have one more axis, which is the replication rate of the virus, okay? Or equivalently, the size, the duration of the responsiveness window. And both of these we can change genetically, so we can get different behaviors. Okay, so that's just one small example, but it's to sort of the riddles that are presented, and some of the approaches we use to look at them. Now, what I always sort of you tell that to people, and if it's a student and they read the paper, they say, okay, now I understand the viral decision, right? But of course, one thing I always have to tell students, you probably are maybe seasoned enough to know that, right? Is that the lesson we use in biology is what we call Occam's rod, right? So in physics, you teach students about Occam's razor, right? If you encounter a simple description for a system, that's a good sign. It maybe tells you it's the correct description, right? The opposite is true in biology. If someone tells you a simple story, that only means that most of the details are under the rug, right? They're either under the rug because we put them there because we wanted to ignore them, or because we just don't know what they are, okay? And so the solution, or the way to move forward in biology, is always to improve your experimental ability so you can challenge those models better, and of course you realize that they're insufficient, they are wrong. Okay, so you have to look under Occam's rod. So the one obvious thing to do is to say, okay, what we did was we did inference of the probability from bulk experiment, population experiment. We've mixed 1 million cells with 1 million phages, and we count. But what does the decision look like at the single cell level? Okay, so that's something we did already. Well, I see it's now 15 years ago. Okay, so we engineered our viruses and our cells so we can look at the decision at the single cell level. Okay, so not going to too many of the technical details, but our phases we engineer so they have a green capsid, so we can see them on the surface of the cell. And in the cell, we engineered a gene expression reporter, so when the dormancy promoter is activated, the cell will turn red. Okay, so in other words, green tells us lysis, virulence, red tells us dormancy. So then we can perform an infection. So here are 4 cells under a microscope. From a big field of view, we'll have hundreds or thousands. You see this cell is uninfected. This guy has 1 phage on it, like this guy, and this cell has 3 phages on it. Okay. So then all we have to do is we let the infection in under the microscope. And so after 80 minutes, you see this cell already is beginning to express the dormancy gene. This one, you see a lot of viruses are being made. By the time you're at 2 hours, this cell has continued to grow and divide. So it's an established lysogen with a dormant virus. This cell is no longer. All you can see are the release of the viral particles. Okay? So then all we have to do is look at a few thousands of these cells. We can measure how many phages infected each of them and what was the outcome. And we get our decision curve at the single cell level. And what that suggests was not what the bulk tells us. Okay, so the bulk inference was a step function, right? 1 phage lysis, 2 or more lysogeny. Now the trend is still the same. When I go to high multiplicity, I approach asymptotically everyone going dormant. But you can see that it's a very gradual curve. Okay, so for example, if I look at multiplicity of 2, I have about 50-50. So in other words, back to the point of predictive power, if you tell me, here's a cell, it was infected by 2 phages, what happened? All I can tell you is, well, throw a coin and make your choice, right? I have no predictive power at this stage, okay? So that is very alarming, okay, at least for me as a physicist. And so what is the interpretation? What causes this variability or indeterminacy, okay? And so as some of you know, there's a very sort of a canonical narrative in the field and in the broader field of cellular decision making, and that is the heterogeneity in cell fate is a reflection of stochastic gene expression. In other words, as it says here, right? There's the unavoidable fluctuations in all biochemical, physical reactions in the cell, diffusion, binding, unbinding, all this chemistry and physics in the cell, and that unavoidably results in fluctuations in the copy number of RNA, in protein, and DNA, and that may end up with noisy, as is the term, cell behavior. Okay, so in other words, in this representation, single cell behavior cannot be treated deterministically, can only be considered probabilistically. Okay, now this has proven a very powerful and popular paradigm or dogma, because if you come like me from physics, or if you come from chemistry, it's very easy to invoke stochasticity. Every single cell measurement you make, you can give a probabilistic description, describe distribution, write a stochastic process. There's a nice pipeline, right? But of course, the problem is that this legitimizes our ignorance, because it tells us, well, what is unknown to you, the single cell behavior, that's perfectly okay because it's really unknowable. Okay, and that, again, so from the physics worldview, this is a bit like quantum mechanics, right? Quantum mechanics tells us, don't worry about what this atom is doing because it's unknowable. Okay, but this is not quantum mechanics, right? Our ignorance can still be reduced by just making better measurements. Okay, cells are not particles. Okay? And so the idea is that, or the argument would be that instead of immediately invoking stochasticity, what we should do is try at least to remove our ignorance by improving our measurements and reveal what quantum mechanics call hidden variables. Variables that deterministically impact single cell behavior, but because we couldn't see them, we called it noise. So that is some of the things that we've been doing. So looking for a clock. Oh, I got it. And I'll show you one, or time permitting, one or two examples. Okay, so let's look at one of these, what was previously hidden from us, which is the step of phage entry, the step where the phage injects its DNA into the cell. Okay, obviously it's a critical step in the infection, and in recent years it's been really pointed out as having very important regulatory effect on cell defenses and anti-phage defenses and anti-phage defenses. It's a very interesting process, and like much of biological and biophysical processes, we very poorly understand it. So we decided to just take a look at it. Okay? So again, the first element is engineering the cells and the phages, so we can perform this measurement. So we combine the things I've already shown you. We have a fluorescent capsid, here it's pseudo-colored blue, and we have a tag inside the phage genome when it comes in. So that means in each infected cell, here's another handsome infected cell, I can count how many phage capsids are on. So here's the E. coli cell in shaded black, and there's 1, 2, 3, 4, 7 phages on it, and 3 of them have already injected their DNA. Okay, so that's all good, and we do this infection in a microfluidic, so I can follow over time arrivals and entries. Okay, so we would get these time series, right? Which we can analyze using standard tools of stochastic time series in many cells, arrivals and injections. Okay. And what we found was something that for really several years we thought was a bug in our experimental design, a flaw in our experimental design, is that the intracellular multiplicity is not proportional, is not linear in the number of viruses on the cell. Okay. So we mix different ratios of phages to bacteria, as in the Kurulski experiments I showed you. When you measure how many on average are absorbed, that's a simple linear function of the mixture. You can even predict a coefficient because it's the kinetics of encounter. It's all very, very nice and simple. But the intracellular number, you see, gets saturated. Okay, you can just normalize it to instead calculate the efficiency. So you have a constant efficiency of absorption that's just about the kinetics of encounter. But then you also have a decreasing efficiency of entry. Okay, and if you, again, same data, but just put all the cells and all the phages together, you can ask, as a function of how many phages are on the cell, how many are inside. Okay, and while when a single phage sits on the cell, it has about maybe 60% of getting in, depending on temperature and things like that. By the time you have 10 or 20 phages sitting on the cell, 3 or 4 come in on average. So there's really a huge diminishing returns on viral entry. Okay, and just thinking about the question of inference that the phage performs on the abundance, you can see how that messes up the whole story. Now, we had to figure out how that happened, and I don't want to get too technical and mechanistic, but I'll say a few words about that. And so first, as I said, from reading literature, we realized that the process is really very poorly understood. Okay, so that's kind of surprising. But there's 2 lines of evidence that kind of led us to form a plausible hypothesis. One is that it is known that the attachment of phages, not even entry, just getting to sit on the cell, creates a huge perturbation to the host. Ions go in, ions go out, membrane potential gets disturbed. Really terrible things without even entry of the phage. And then we also know from in vitro studies that ionic conditions should also impact the ejection. And so putting those together, it was easy to hypothesize at least that attachment of phages creates an ionic perturbation in an MOI-dependent way that then impedes entry. Okay? And then we tested this, and that indeed proves to be the case. Okay, and so one way you can look at this is adding to our phage indicators an indicator of the membrane permeability. So that's a handsome purple dye. It's rubidium iodide, tells you how permeable is your membrane. And so when you look at your infection data, there's a very strong correlation between how many phages are on the cell and the permeability of the membrane. Okay, but again, this is not about the phages coming in. There's no correlation with the intracellular number. This is just about absorption. So it's enough that the phages attach to the cell that they make conceptually a bunch of holes in it that leads to permeabilization. And then if you analyze the same data using what's called causal inference, you can indeed find that permeability impedes entry. Okay, so more phages, more mixing up of the membrane, harder to get in. Okay, and another way to look at this, which I'm mostly showing because I like the essay, is by looking at what it does to cell motility. So some of you may know that cell motility, bacterial swimming, also responds very strongly, really is an indicator of the membrane potential, the proton motive force of the cell. And so it actually can be used, if you follow cell swimming, you can tell how much energy is in its membrane. Okay, so here with the collaboration with my friend and colleague Jan Schemla, we do this in an optical trap. So we have, it's going to take forever. So this is an optical trap. I mean, this is a microfluidic device, and there's an optical trap, a laser tweezer that holds the cell in place. And so the cell is swimming, but it stays in one place. We call it the bacterial treadmill. Okay, so it runs and runs, but stays in one place. And then we infect it with a fluorescently labeled phage. Okay, so the cell is swimming, so the phage on the cell keeps going up and down, because we're looking for the side, and this is a readout of the cell rotation. But in all these infected cells, within a minute or two, they all stop swimming because the phage infection depolarizes the membrane. It takes away the proton motive force. This takes in about, movie's too long, but in about 5 seconds, I think. Doesn't happen, I'll play forward. Okay, stop. And so you look at many cells and they all do that, or all but 1 or 2, a few percent, okay? They stop swimming. And if, of course, you take uninfected cells, they're happy to keep swimming for an hour. So again, another way in which the absorption by the phages completely de-energizes the cell transient. It then recovers. Okay, and that in turn prevents or at least slows down the entry. So the bottom line is that you have an MOI-dependent perturbation to the physiology, electrophysiology, that then impedes the entry. Okay, now the question is, does it make any difference? What are the consequences? And there's at least 2 that I want to mention. One is, again, in the context of the propensity to become dormant, right? So as I said, the common interpretation is that the phage infers the abundance, the relative abundance of phages to cells, and based on that makes a decision. And so if you consider now the fact that how many get in, and so therefore how many copies the circuit actually has to count, depends on the ionic and chemical environment, that propagates into the decision. So here's a simple demonstration of that. So we have 2 different common media that are used in phage experiments. One of them is the one I've been using, and so there's this sort of gradual decrease in phage efficiency entry. And here's another media, again, a very common one, that even if you have about 10 phases on the cell, on average, barely one comes in. Okay. Now you take these two media, and again, they're very standard, very commonly used, which is what messed up things for us for a few years. And you say, well, I expect I'll have much fewer lysogens here, given at the same mixing, because even if I put two phages getting to on the cell, most of the time it's just going to one come in. So there's not going to be lysogeny. And indeed, this is what happens when you perform the lysogeny experiments and you scan the ratio in the mixing. There's about a 30-fold difference in the maximum probability of dormancy in the what we call a poor entry medium. Because while you have multiplicity of almost up to 100, most of the inside of those cells contains only a single phage. So the circuit counts and says one phage, go lithium. Now, another consequence is regarding the heterogeneity. Okay, so the question is the following. If I don't want to invoke stochastic gene expression, if I look at the decision circuit that I inferred, maybe 10 slides ago, and I treat it as completely deterministic, the decision circuit is, ODEs, just deterministic. But I do allow for the fact that the entry has this temporal kinetics. Is that enough to create heterogeneity in the outcome? And the answer is yes. Okay, so the black data is the single cell data I showed you a few slides earlier. And the blue is what the ODE model gives with a naive picture of entry. Everyone on the outside immediately gets inside. But if you take the stochastic entry that we inferred, the entry kinetics that are, what's the word? I'm blanking on an English adjective. Okay, that is gradual and imperfect. Then you get already a considerable cell-to-cell variability, right? Because even if there was two phages on the cell, maybe one got in after a minute, the other one got in after 5 minutes, or never got in, and so that would propagate into the decision. So in other words, while this did not completely remove the need for randomness in the decision circuit, it removed a lot of it. Okay, so even if we treat the decision circuit as fully deterministic, we got already a considerable amount of heterogeneity upstream of that, if you. Okay. Okay, I'll try to see what I can get. So let me just say a few words about what else is in the decision. So here's a reasonably up to date, well, 10 years old cartoon of the decision circuit. Okay, so we only looked at C2, C1, these guys here. But even from what we know, and of course we don't know, the decision circuit is much bigger. So there's yellow are viral genes, blue are host factors. And so you can see there's a lot of input that comes from the host, okay? Including several key players that are known to respond to the growth rate and the metabolic state of the host, okay? So very naturally, when the virus comes in, it also senses, and perhaps it's the most important thing, it senses what is the state of its host, how healthy, how fast growing is the host, okay? So you can easily imagine, even again, just taking this decision circuit and plugging into it the textbook picture of how gene expression parameters change with bacterial growth rate. This is that axis. Right away, you will get a two-dimensional decision curve that would have different regimes. Okay? So in other words, even just taking the most basic textbook knowledge of, again, E. coli growth rate regulation, you already get behavior that we didn't see before, and that you can imagine that too would contribute to heterogeneity now, because if in the same experiment the cell I infected was growing 5 minutes faster because of where it is in the agar or something, I may get a different outcome. So that's another possible source of heterogeneity. But the challenge is that our understanding of host regulation of the decision is really very poor. Quantitatively, it's close to nothing. So we wanted to start looking into that. Now, technically, now we need to scan multiple sort of input parameters, host growth rate, temperature, carbon sources. The single cell assay is not really a good assay for us because it's a very low throughput. So now we're going to go to the side of high throughput. And so we devised, we go back to the population level, and we devised a high throughput experiment. So this is all in a plate reader. Some of you may have used those, so you can do 96 or 48. And we found that we can have good reproducible infection kinetics, and we can use them to infer some of the critical parameters. let me just show one or two examples. So we have a simple model, and the model is very similar to things Marianne was showing yesterday, and things you would find some of them in the textbook. And we can use this model to interpret. So here's a typical infection experiment. We have different multiplicities, and what we're measuring the data over time is the bacterial number, okay, which in the plate reader you can measure automatically. So these are different curves, the different infection multiplicities. And the dots are the data, and the curves are the fitted model. Okay, so that's nice, but again, fitted model is is what we do for a living, but you shouldn't immediately be impressed because maybe you had enough parameters, and you could fit the elephant, as they say. But the nice test that this is useful is we can then use this to predict the phage kinetics. So here again, data is the dots, and the curves are the model, but now this is not a fit, this is a prediction, okay? So we did fitting here and predicted what happens to the phage. And you see it's very reasonable. So without making any measurement on the phages, we can predict their kinetics very reasonably. And so that means we inferred, or suggest we inferred parameter reasonably. And as always, as often in models of biological system, it's not that the models are individually well constrained. The models are what we call sloppy, but if you can identify what model is, what parameter is what we call stiff or very narrowly defined, you identify this rate of viral reproduction, and also you find that it grows more or less linearly, or at least monotonically, with the growth rate. Okay, so one thing we already inferred from here is that as the growth rate increases, you get more viral reproduction rate, which is of course very intuitive, but it's nice to get that from the data. Now, similarly, we can use the assay with a slight modification to measure dormancy. Okay, so now all we're adding, the infection is the same, but we're adding the antibiotic selection, which you remember is how we count lysogen. And so then we can, at different multiplicities, the cells will start growing at different times, and we can extract them and ask how many lysogens I had. And we can again get these lysogeny curves, but now you just see we can get many more data points because this is done in a multi-well, everything is higher. Okay, and so one of the things we could do is ask what happens to the propensity to go dormant as a function of the growth rate. Okay, so here we have the bacteria are growing, and different times in the growth, different stages of the growth, I take them out and perform a lysogeny experiment. Okay, and so it's just something that you could do in principle with plates and everything, but it's just so overwhelming that it's much easier to do in a plate reader in a high throughput. And making a long story short, there's a very clear trend that as you increase the growth rate, the propensity to go dormant goes down. Again, the data is actually quite dirty, it's not very good, but the trend is quite clear. So when cells are growing faster, there's a lower propensity to go dormant. You make more viruses if you go litty, you get higher reproduction, but you also get, at the same time, you get lower propensity to go dormant. Okay, so we're still looking into that. I think it's very interesting. Okay, let's see how I'm doing on time. I'm 1 minute from my 45. So let me see. Okay. I'll just very briefly, because I had some nice data, but I'm not going to cite it tonight. So one of the things we found is that if you look at the decision curve at the single cell level, okay, and now you consider not just the multiplicity, but also cell size. Okay, so we had a very strong prior what to expect. We thought we would have a dependence on the concentration of phages, right? So one phage infecting a cell of this size should be the same as 2 phages infecting a cell of this size, because the concentration of the phages would be the same. But instead, in our analysis, the scaling analysis, we found that there's an extra power of m. Okay, an extra power of the multiplicity. So there's an extra dependence we didn't expect, and the way we interpret it is just from the phenomenological point of view, from the math point of view. What will give us that is that the viruses first make individual decisions, and only if all of them choose lysogeny, this is where the power n comes from, you will get lysogeny. Okay, so the math works very nicely, but of course the mechanistic question was for us very odd. How can phages maintain individuality? What does it even mean for a phage to make an individual decision where you're in a 1 micron cubed volume where everything gets mixed within a sub-second? Okay. So how do they maintain individuality in a shared cytoplasm? And so we don't have the answer, but I will just tell you some of the enabling technology we use is the following. One is we have to look at, so we label now multiple genes from the decision circuit simultaneously. And so that also allows us, so here's one cell, again, this is the reconstruction, to identify the transcriptional activity of the individual phages. So now this is reconstruct, this is the RNA for different genes from phage 1 and phage 2. So now in the single cell, when I look at the expression of the genes, I don't look at the whole cell. I look at what virus 1 is doing versus virus 2 versus virus 3 over time. So that's one window into that. And the other window is that, you know, if you do standard optical microscopy, some of you may do that, you know that your resolution limit is mostly almost the size of E. coli, right? So to start looking at things more finely in E. coli or in any bacterium, you got to increase your resolution. So now we've gone from, this is traditional microscopy images I've shown you, to what is called min-flex microscopy. Where did I write that? So that's a nanometer scale imaging modality, and it's very exciting. And so now the DNA, instead of looking in the capsid, instead of looking like a diffraction limited spot, you can actually begin to see the conformation of the DNA. And when you look inside the cell, instead of seeing a red blob, you actually get to see these are individually labeled nucleotides. You actually get to see the polymer nature of the DNA. So this is the edge of the cell where the DNA is coming in, and you can begin to see the actual DNA conformation. So we're getting into the nanometer scale of things. Okay, and this is probably where I should pause. So let me just remind how I go to the thing. So I will go here. Okay, so what we want to get with this is really that our decision model will be spatially informed. So we will involve the individual copies of the phage and whatever happens is spatially inside the single cell. Okay, and let me just repeat very briefly my take homes. One is, again, I find it an excellent test bed for, you know, for our failure to predict biological behavior. Two of the caveats I've mentioned, and really it's a bit of a trivial lesson, but at least in our hands, it's proven very helpful and informative to zoom all the way from population to the single cell and the single phage sometimes. Okay, and this is probably where I should end. So I will thank the people who do the work and some people who give us money, and thank you for your time.
Thank you very much, Sido. Thanks for also respecting the time.
I hate people who work with time.
Yeah, that's simple. The floor is yours, and also the people online, you can leave your questions in the chat, please.
Yes, Yoshi.
I want to understand more the last, the first part. In the last part, you show the stage of injection, spot, and what is the plus axis?
Sorry, tell me which. Here, let's see if we do it like this. Which slide am I going to? Can you tell? Here, let me make it a little bigger. Which part am I going to? Remind me. Can you tell me?
The first part.
First part, okay. Is it this one? 15?
Yeah.
15.
Yes.
So in this case, the x-axis is multiplicity. The y-axis is the rate of viral replication.
Okay.
So in other words, the doubling time of the phage inside the cell. So this is the non-replicating mutant, and this is approximately the wild type, and you can have a yes.
Do you assume the degradation rate
Of
the phage?
no, the phage DNA does not. I mean, I don't want to say it does not, but in a healthy replicating phage, they have defense mechanism from lambda from preventing DNA replication. But of course, you have dilution, right? If you work in ODEs of concentration, the cell is growing, so the concentration of viral DNA constantly decreases.
So that is the reason for the phage infection. So.
Oh, I see what you're saying. Where it was that? Yes. So, right, you have constant dilution of protein products and DNA concentration. You're right, and so you have to compete with that. That is true. So even if the cell is not dividing, you will see an infection that the cell is constantly making new mass and elongating. That is true. So you need that to get to any asymptotic behavior, including the failed infection. That is correct, right?
And in another question, in the last part, you show the spatial localization for different pages.
Right. Let me.
What's the mechanism of the-
Sorry, where am I? You mean like this, for example? Yeah. What is the question?
What's the mechanism of the separating the localization? for different-
Well, I mean, in this case, for example, these will really just be two phages that came in. So of course, they come in at, you saw in the picture, they come in many different places. So they don't.
So, okay.
Now, what it has been shown that when they come in and they replicate, they typically tend to keep their offspring copies in the same area. Because what you don't see here is, of course, that here in between them, and you can almost conceive it, is the bacterial nucleoid, the genome. And so that is a strong hindrance to things moving around the cell. So small molecules can go, but big objects like phage DNA is typically stuck on the other side of it. Yeah.
Okay, thank you very much.
You're welcome. Yes, sir.
Yeah, that's all super cool. And I like the argument as well. And the stochasticity, and the question I had was, when you have MOI on your X, is it the true MOI because you can measure it, or is it the ratio of what you throw in?
So it depends, right. So that, in a way, our progress over the last 20 years is in improving what we put on the x-axis, right? So the first experiment, a la Kurilsky, is really mixture in the population. In the, let's call it 2010 version, it is, let's see for a second, 2010 is this.
Yes, the flick, exactly.
So 2010 is phages on the cell. Sorry, I should have been, is on single cell, how many are on it? But as we saw a few slides later, that is not how many are in it. So what we have not yet been able to do is, if I really measure in live cells, how many are inside versus the outcome, how close is that to a step function? So we're getting close, but that the dream plot that I keep promising in proposals and things like that is that if I identify this axis correctly, I will get experimentally a step function. May take me another 20 or 40 years, but that's what is that on the scale of science?
So you could still have the MO1 with 2, you could actually still have a certain number of ones. But
in this one,
stocasticities, so that you
in the entry, is in the entry process. Right, and that was what at least our inference from the later part suggests that is an important contribution. So in other words, if I, even numerically, that's what we did, I take these values and I have my inferred model for entry, which we get from this microfluidic data. And so now I can convert that, although still stochastically, into how many are inside, exactly. And the data is consistent with the fact that if I was able to recalibrate this to the inside, it's a much sharper curve. It may not be a step function, but it's got a hill of about 10 or so. It's a very strong, much less stochastic or much more precise curve. Exactly, correct. Yes.
Yeah, I had a question on one of the slides you showed back, following Rich's question. Let's say that when the MRI gets above a certain threshold, you have a mixed outcome in the model, and I think I missed that some.
So I probably did not say anything smart about it, at least. Yes. So first of all, to some degree, in the model, the way this comes about, and of course, the model is, even by design, is a very primitive model. There's a lot of things that are not in it. I think this mixed outcome is when you reach both thresholds within the time window that you define for the decision. So within an hour or something, both the lytic gene and the lysogenic gene reach a threshold. So of course, not of course, but presumably something happens, the cell does something in response to the phage, in our model, the model is dumb enough that it just calls it mixed. Now, it is interesting that it is known, and it's even in the data, when you go to very high multiplicities, you start seeing reduction again in the number of lysogens. I'll show it in the, let me show it a second. It's in one of those groups I can see very clearly. Let's see these ones. So these are different lysogeny curves, and very often if you get to very high MMOI, you start dropping again. So we don't know what happens to these cells. Is it just death like lysis, or just death without productive viral production? It's very easy to imagine. I mean, that if you put 20 copies of lambda in the cell, some of the genes will just become lethal in an unhelpful way. So in other words, you're not going to get productive, successful reproduction, but you've done enough to kill the cell. Okay. Now you can even, if you think more strongly about ecology or evolution or the optimality of things, you can say, oh, that's why the phage impedes the entry. So even if 10 phages are sitting on the cell, don't let more than 3 come in, because then you're just going to make a mess inside the cell. So you can kind of argue it different ways, but sorry, yeah, sorry. There was a sorry.
So at some point you talked about the counting of the exchange of certain time, and there's also an influence of whether the 2 phages were fixing the same time. And then later in your talk, when you refine your definition of MOI, this notion of simultaneity or separated in time disappeared. Did you try to figure?
So the way in my mind it gets reconciled is that your window of counting is such that that window does account for or allows for the replication. So when phages come in, if they're wild type, they would always replicate. So in other words, the counting is effectively of how many phages are there by 20 minutes, let's say. And so that does involve the replication that always happens. So in other words, if 2 phages come in, by 20 minutes, let's say you have 8 copies, that is the number that gets counted in threshold. Okay, and that is why if you have a non-replicating phage, the 2 phages that came in, they stay 2, that does not give you lysosome. So in other words, the counting, as it happens already, takes into consideration the way it's calibrated, obligatory replication within 20 minutes. So that's how sort of I consolidate it, or how I accommodate that. Okay, so the counting, the phage does do an integration, and that integration assumes, let's say, assumes constant replication. So the counting only happens until there's a decision. Once you have a decision, then if the numbers go up and down because of these confusing factors, that will not impact the decision. So that's sort of the picture. But it's right, it's not perfectly binary. It has heterogeneity or analog element. Sorry, Vasily, you were saying.
Yes. So back to the previous point with the mixed results. So actually, what it means from the model is like that the concentration of these 2
proteins, right?
Proteins are on similar levels.
Right.
So if you can you expand a little bit on this? What, as you said, like it goes down the resource and so on. What would that mean then? Like what do you imagine this happens actually?
So one thing that can happen, and is anecdotally seen in single cell experiments, that you get an infection where the cell stops growing, but you do not get successful dormancy or release of viral particles. So everyone loses. So there's maybe 100, let's say 100 phages in the cell, a lot of phages in the cell. They make enough, because remember, a lot of the things that the phages express, they modulate host functions. That's what they do, right? Because the phage comes in and needs to hijack all the cell machinery for making DNA, making RNA, making protein. So you have enough phages, so they hijack the cell is not going to grow and divide. But because of where they are in the copy number, they've also messed up the decision, and they're not. So you have these high numbers. By the circuit, you should go dormant. But you've expressed so many things, so much high levels, that you've killed your host or immobilized your host, and so you end up with nothing. Under a microscope, you will see not successful production of phages, but also not resumed cell growth. So it's
basically it's not a mixed result. It's
right.
a new state basically.
Yeah, exactly. So exactly right. It's called a mixed result only in the context of the very limited model. It only knows did I hit the right wall first or the top wall first. If I hit both of them, the model says outside what I know. Exactly. And so then you can always invoke a new interpretation, but we didn't have data to support it, so we just left it as. But you're right. It's only the model definition is mixed. It's not a. The biological statement is not that, or is different, right? You're the chair, I think. So
Yes, I will take this the last three questions. Yes, Clara.
Just at this very high MOI, would you consider the lysis from without the phenomenon plus the physical?
Yeah, so I think that in Lambda we don't have evidence for that. Even again, that also goes with the cells question. So even in Lambda, there's all these very, it's never really binary. So one of the things we've seen, so lysis from without we do not see in Lambda. One of the things we do see in Lambda is a episomal state. In other words, the phage stays dormant for a while, but is not integrated. Okay, so another thing you see, I was talking to Silvano, that is you see multiple copies integrating as lysis. So you do see all zoo of behaviors, but we do not see. It doesn't mean it never exists, but we've not seen lysis from without. Sorry, in Lambda.
Diego. Yeah.
I was wondering for the movement, changing movement of the bacteria. Does it display like differability of encountering other phages, or it actually makes the bacteria an easy target for?
That's a very good question that there's been theoretical studies for 20, 30 years, going back to really some very fundamental biophysics theory. It's unresolved. There was just a recent paper, theoretical, that looked at did a more detailed simulation of the boundary layer around the swimming bacterium from the fluid mechanics point of view, and argued one way or another. Theory-wise, there's argument both ways. It's on our list to look into that. Haven't got to it, but it's a very interesting question because, right, I mean, phage infection and chemotaxis or motility is two basic elements of bacterial life. And so how they interact with each other is absolutely very interesting. And after that, in terms of metabolism, they're both very costly. So it's a very interesting question, I think.
And the last one is.
In the last part of this pathology, we prefer to assume that you expect that the close proximity of where the phage are infecting is gonna have an impact on the lysogeny rate, and if there are known structure to how phage absorbs already. I don't remember if it's lambda that has something to do with the poles.
Yeah, so one is, as you say, correct. So for multiple phages, it's been shown that phages prefer to enter at the poles. Interestingly, we see it in some conditions, but not others. So it's not, and it's been suggested, or I have not seen evidence that has consequence for the decision, because in E. coli, different proteases, RNases are localized to the poles. And so it's been argued or hypothesized or speculated that results in effect on the choice plasminogen, because they go in the poles where they would meet more or less of the degradation enzyme that would result in more. From the limited data we have, we've never seen spatial impact on the outcome. It doesn't mean that it's not there. It could be that we need better experiment, but it's a very interesting, as a speculation, it's been brought up by multiple people. So I think it's an interesting idea. I just have not seen the data to support it.
Thank you, everyone. Also, thanks to the people online. Thank you, Igor.